Good day, class. Welcome to our revision end of year grade 11 life sciences. Please note that our papers that we are going to write end of the year, I'm giving you a replica, I'm giving you comprehensive revision here. Please keep following, don't make mistakes. The year, end of year is soon, is now, and please don't fail. Well, its metric up is there. Without wasting any time, note that this is a 2020 paper, and then it's an exemplar, paper 150 marks. I'm not gonna read any instructions. I'm sure that I once read them before in the previous paper, so let's just go to the questions so that we don't waste any time. We go straight to question 1. A common function of calcium and phosphorus. A common function of calcium and phosphorus in mammals is that it dash. A common function of calcium and phosphorus in mammals is that it forms part of nucleic acid, not really. Plays a role in the synthesis of proteins, not really. Calcium, it is known for protecting. It actually prevents rickets. The bending of the bones, it strengthens the bones, so it prevents rickets, which is C. And then next question, which of the following substances are absorbed by root hairs from the soil? Which of the following substances are absorbed by the root hairs from the soil? Which one of them is absorbed by the root hairs from the soil? We have got 1, mineral ions, they are absorbed. 2, we have got nitrates, they are absorbed. Oxygen is not absorbed from the soil. We have got, we are saying oxygen is not absorbed from the soil. Obviously, we also have humus, it is absorbed. And then we have got water is also absorbed. So I'm saying it should be 1, 2, 4, and 5, which is letter B. And then B is the correct answer on question number 2. It is only mineral ions, nitrates, humus, and water that are absorbed by root hairs from the soil. In which of the following processes is mitosis not involved? In which of the following processes is mitosis not involved? Production of sperm cells in the testes. Obviously, this is the right answer here. It is A. Number 3. A is the answer. We cannot go anywhere further. Gametes are produced by meiosis class. Understand that meiosis is the one that produces gametes, which is mitosis does not do anything in the production of gametes. It is for cell replacement and cell elongation and growth, basically, to say that is mitosis. And then production of gametes, which is sperm cells or ovules, it is done, or it is a process that is promoted by what? By meiosis. So the answer is A, not involved. It is A. Question, we are having 1.4 based on the diagram showing the cell placed in salt concentration. It is placed in salt concentration, and then we have got this given 1, 2, 3, and then the diagram above represents dash. The diagram above represents dash. What is this diagram representing? I'm sure you remember the process of plasmolysis, which is actually the one that is shown here. We are saying the diagram represents what? It represents plasmolysis, which is letter C. It represents plasmolysis, which is shown by this, where we are saying it is a cell that is placed in salt concentration. And then by end of the day, we will be finding the changes that would happen to this cell. So it is plasmolysis that is shown there. How will the water how will the water potential at 1, 2, and 3 compare? Number 1.5. How will the water potential at 1, 2, and 5 compare? how would the water potential compare at 1, 2, and 1, 2, and 3? So with this, you should understand that the answer should be that 1, 2, and 3, the water potential, we should understand that is water potential at 1 is higher than at 2, and 2 is higher than at what? It can be true that to say that you can see that the water potential at 1 is higher than 2, and then at 2 is higher than 3, which should be understood that in each of these levels, 1 is higher than 2, this one is higher than 2, and then 2 is higher than 3, is higher than 3. So what is happening is that 1 is higher than 2, and 2 is higher than what? Than 3. That is what is happening. So please note that 2 is higher than 3, 1 is higher than what? Than 2. That is the the water potential difference, which is given that the answer is what? Is D. D is the right answer, which is that water potential at 1 is higher than at 2, and at 2 is higher than at 3. Tomatoes tomatoes are red because of what? Tomatoes are red because of dash. Number 6. Tomatoes are red because of what? Because they have got what? They have got many the answer that in number 6, it is because they have got many chromoplasts that are formed there. We are saying tomatoes are red because they have got many chromoplasts that are formed. Answer D, number 6. And then number 7, which property of enzyme is represented by the sequence of diagrams below? I'm sure that you remember that enzymes, they act on specific items, and then they catalyze, they increase the rate of reaction, they speed up chemical reaction. So basically that is the effect of enzymes. So, and also you must understand again that the enzymes are also specific in their function. So in this case, what is being shown in this diagram, it actually identifies that enzymes are what? Are specific in their function. They don't just work randomly, but they work specific in their function. The diagram is illustrating that enzymes are specific in their what? In their function, which is letter D. Enzymes are specific in their function as being represented in the sequence of the diagram that we are seeing here. To see that, you can see that it's a key and block reaction, which shows that the enzymes are specific in their reaction, in their function. Number 7 is D. Number 8, which of the following are characteristics of electrocytes? Number 8, which of the following are characteristics of what? Of electrocytes? A, they contain electrocytes. We are saying A, they contain hemoglobin. That is true. They've got red pigmentation. Also, they destroy bacteria. Not really. And then we also say that assist in blood clotting. They assist in blood clotting. Not really also. It is not true. And also produced in sponge tissue of the long electrocytes are produced in the sponge tissue. That is also correct. So our answer number 8 is what? Is A. A is the right answer, that they contain hemoglobin and are produced in the sponge tissue. Number 9, the table below shows differences between plant and animal cells. Which comparison is correct? It is a plant cell and an animal cell. Which comparison is correct? Is incorrect. Yeah, we need to check the questions very correctly. It says incorrect. It says incorrect. Which comparison is incorrect? Large vacuum, and then the vacuum is smaller or absent. That is true. Number B, cell membrane present, cell membrane absent. Which comparison is incorrect? we are saying the answer number 9, obviously, it comes to say that the cell membrane is present in both plant cells. The cell membrane is present. So our answer is what? Is B. So our answer here is what? It is B. Because I'm sure that you know that plant cells, they know they don't have chloroplast. The fiber cells, they don't have, which is D and all other things. They are correct. And then number 10, the diagram, study the diagram below, and then the function of the root, the function of the plant organ in the diagram above is dash. The function of the plant organ in the diagram above is dash. What is the function? It is what? What is it responsible for? I'm sure that you can see that this is actually, which is it's responsible for increasing the surface area for absorption. I'm sure if you can refer to your notes, you can see that it is specifically responsible for increasing the surface area for what? For absorption. It increases the surface area for absorption. Number 10, the function of the plant organ in the diagram is to increase the absorption area for absorption. And then we give the correct biological term. Give the correct biological term in each of the following descriptions. So carbohydrates made up of 3 or more monosaccharides. What are they called? We call them what? 3 or more monosaccharides. We call those are called what? They are called polysaccharides. They are called polysaccharides. They are called polysaccharides, a chemical element in hemoglobin that is essential for the transport of oxygen. A chemical element in hemoglobin that is essential for oxygen transportation. That is called what? It is called iron. I'm sure that you remember that it is iron, which we can give as what? It is called iron, which I'm sure that you remember that iron is the one that is found in the hemoglobin that is essential for oxygen transportation. Number 3, wax layer that covers the leaf, preventing excess loss of water vapor through evaporation. This is called the cuticle. It's like a plastic cover that is found on the leaf structure, which is actually there to avoid or to reduce water loss. It reduces the evapotranspiration. It is actually responsible to enable plants to survive in dry seasons or during dry times. So it is called the cuticle. It is called the cuticle. And then an opening through which the spinal cord leaves the what? The skull. I'm sure that you remember, if you look on the structure of the skull, where the skull connects to the body vertically there, or that is for bipedal, and then horizontally on quadrupedal, I'm sure you remember the foramen magnum. The foramen magnum, it is actually the opening through which the spinal cord leaves the skull. A blood group known as universal donor, it is group O. When we group blood in terms of ABO blood grouping, when blood groups is done through ABO blood grouping, we have got blood group O, which is recessive. The thing is that it contains no antigens. No antigens are found in blood group O. So it is called universal donor, because other blood groups, which is AB or B, they produce specific antigens that are also a problem when they are given to people who don't have that blood group. Use of chemical agents to treat cancer, it is called chemotherapy. It's called chemotherapy. The chemical agents to treat cancer is called chemotherapy. And then fat mainly derived from plants. Fat mainly derived from plants uh, and are liquid at room temperature. They are oils, and we can quickly just agree that these are called what? These are called oils. And then the type of tissue in the vascular bundle causing secondary thickening, type of tissue in vascular bundle that is, causing secondary thickening, number 80, we call that what? It is called the capium. It's called the capium, which is actually a tissue in the vascular bundle causing secondary thickening. And number 9, collection of cells of the same type performing the same function. We call that a what? It's a tissue. Number 9, it is what? It is a tissue. Collection of cells of the same type performing the same function. We call that a tissue. The loss of water in the form of vapor through aerial parts of the plant is called transpiration. That is called transpiration. Transpiration, that is the loss of water vapor in the form of what? Of the water vapor through the aerial parts of the plants. When water is lost from plants, we call that transpiration. And please don't confuse it with what? With when it is lost from water, is lost from plants and water sources. Is the combination of both. Is called evapotranspiration. But if it is plants alone, it's called transpiration. Number 1.3.1, reactions in which enzymes are evolved. Reactions in which enzymes are evolved, we indicate that is A, B, only, or both, or none. Number 1.3, reaction in which enzymes are involved. Enzymes are involved in what? In catabolic reactions. It is also involved in what? In this. So it is both A and what? It is both A and B. And then the medium in which chemical reactions take place in the body, the medium in which chemical reactions takes place in the body, chemical reactions, they take place through what? The medium in which chemical reactions takes place in the body, they take place through, it is through the enzymes only. A only. And the scientist who invented the electron microscope in 1939, this guy was called Max Nott. It's called Max Nott, B only. And then we can go to the next question. Is the diagram that represents a sign of a sign of a joint, synovial joint. A synovial joint is shown there. And then the question is saying label part A and B. Label part A and B. So A, we can label A to say that our A, it is actually a joint capsule. This is called a joint. A is called a is called a joint capsule. And then obviously you should understand that B is the cartilage. You should understand that B is the cartilage. And then I saying label A, parts A and B, it's actually a sign of your joint. So A and B is showing the capsule. B is the cartilage and A is the joint capsule. And then identify the synovial joints illustrated in the diagram above. Identify the synovial joints that are illustrated in the diagram above. question 1.3, 1.2.4, 1.4.2. This joint is actually called the hinge joint. It's called the hinge joint. That is the synovial joint that is illustrated there. It's called the hinge joint. It's actually called the hinge joint, which is actually illustrated in this whole diagram. So it's called the hinge joint. And then next question is saying, explain what would happen if fluid in the cavity C dries up. In the cavity C, the cavity C, it actually contains a lubricating fluid, which needs to make sure that the cartilage does not go through friction. So if the fluid dries up, explain what would happen if the fluid dries up here. Actually, the fluid lubricates the joint and the cartilage. The fluid, it lubricates the joints and cartilage. That is the importance of the fluid there. Actually, the reason for the lubrication, it lubricates the joints and the cartilage to prevent friction. I'm sure that we understand that. To prevent what? To prevent friction. It lubricates the cartilage and the joint to prevent friction, and also, which is also, when that is also, we are saying friction and what? And drying out. Friction and drying out is prevented by the fluid there. So I'm sure actually it makes sure that the friction does not take place because of the drying out. So drying out of the fluid results in painful, also difficulties in mobility as well as inflammation. Drying out of the fluid, uh, it results in inflammation. It results in what? In inflammation, or we can say inflammation results in inflammation. It results in inflammation, which we can also talk about, also difficult inflammation and difficulties in movement. Inflammation and difficulties in movement. Difficulty in movement. We are saying the point here is that fluid that is contained there, it actually lubricates the joints and cartilage to prevent friction, and drying out also just to make it, to keep it lubricated. So drying out of the fluid results in inflammation and also difficulties in movements. That is one thing that you must understand with regard to the drying off or drying out of the fluid that is contained in part C. And then name two diseases that can affect joints. Question 1.4.4. Actually name two diseases that can affect joints. One, we can talk about there is what is called osteoarthritis. One, we have got the it's called osteoarthritis. It's called osteoarthritis. Two, We can also talk about the rheumatoid arthritis. These are the two diseases that can affect the joints. Is osteoarthritis and then the rheumatoid arthritis. So those are the two diseases that we can talk about. Those are the two diseases that we can talk about. Let's quickly just go through question 1.5. We have got complete the table that is based on chemical tests for organic food compounds. Organic food compounds chemical reagents change for a positive result to show that that is present. The change is shown there. So question 1.1, first question is starchy chemical reagents. What we use to test for starchy, I'm sure that we can agree that is iodine. Iodine solution is the one that is used, and then it turns from blue to it turns blue black. It turns blue black color. Proteins. What is used to test there? We can use copper sulfate. We can use copper sulfate here. We can use copper sulfate. Proteins can be tested with copper sulfate, and then also the color changes that can be there. The color changes when we test. We can have brick red. We have got brick red or violet, which changes to brick red. We can say brick red, or it can also be uh, violet. Brick red or violet. And then the next question is saying number 1.5.4, we have got the chemical agents is ether, and then 1.54, we can talk about fats or lipids. Fats or lipids, we can have fats, or we can say lipids in this case. And then also in terms of the color changes, at number 1.5.5, the color changes, it can also be brown, or also it can also become, we can talk about brown. We can have brown, or we can have filter paper, which we can say again, maybe we can also talk about the brown, or the transient stain on the blotting, or also we can also talk about the filter paper there. We can talk about next question 1.5.6. It is orange to brown. Orange, I mean, it is organic is not known, and then it turns orange brown color results. What is that? We can say number 6. It can that is we can talk about simple sugar, which you can say glucose. You can say glucose here. Glucose. And then number 7, which we can say what is the chemical agents that can be used there. We can use also Benedict solution. We can use Benedict solution. can use vindictive solution, which can also be used there. We can also, certainly, we can as well use, still on the same answer, we can also using the fillings A and B. They can as well be correct answers on what? On question 1.7. Question 1.7, we are saying vindictive solution or fillings A and B. They can as well be used in terms of glucose test. Next question, which is 2.1. It is the extract and answer the questions. Sometimes during cell metabolism, chemical substances which are poisonous to the body are formed. However, the body cells are not destroyed due to the presence of enzymes that decompose these poisonous substances into harmless products. They are decomposed into harmless products. One such poisonous substance that is released as byproduct during normal cellular respiration is hydrogen peroxide. However, this enzyme catalyze ensures the enzyme's catalysis it ensures that this poisonous substance is converted into two harmless substances or products as quickly as possible, which is hydrogen peroxide is converted, and then it is actually broken down to have water and oxygen, which are given there. And then the question is saying, the first question is saying, what is the function of an enzyme? What is the function of the enzyme? It's actually a biological catalyst that speeds up a chemical reaction. In this case, it is a biological catalyst. This is it's a biological catalyst. It's a biological catalyst that speeds up or accelerates what chemical reaction. Catalyst that speeds up, or we can as well say accelerates chemical reactions. It accelerates chemical reactions. That is the function of an enzyme. In this case. It catalyzes, it speeds up or accelerates chemical reaction. And then next question is saying, describe the benefits that humans derive from the function of enzyme mentioned above. Obviously, it is mentioned that it helps the poisonous substances. Poisonous substance is converted into harmless substance, which prevent poisoning of the body tissue. That is what is said in the essay. It actually, poisonous substances are made harmless. Poisonous substances are made, they are made harmless to the body. So it is actually helping to preserve, to protect the body. Chemical substances are poisonous substances or dangerous poisonous substances are actually converted into harmless substances which prevent poisoning of the body tissues. Body tissues are not poisoned. I'm sure that we have seen in the essay there. And then question C, identify two harmless products released during decomposition of hydrogen peroxide. These are actually given here. When hydrogen peroxide is decomposed, we have got oxygen and water, which is given there. This is where it is shown. I'm saying This one is actually shown where it is given that it is going to be broken down, and then we have got what? Carbon dioxide, and I mean we have got oxygen and water. So it is what? Water and what? And oxygen. It is water and oxygen that are the two harmless products that are produced during the decomposition of hydrogen peroxide. What effects does the involvement of an enzyme in a chemical reaction have on its structure and its ability to take part in the subsequent reactions? Actually, you can see that in this regard, we can understand that the enzymes remained unchanged and can be used repeatedly. It remains it remains unchanged and can be used and can be used and be used repeatedly. That is true. It remains unchanged. It actually remains unchanged, class. It remains unchanged and can be used repeatedly. That is the issue in terms of the enzyme in a chemical reaction with regard to its structure and ability to take part in the subsequent reactions. And then question question one point next question, which has to do with an experiment to determine the effect of different temperatures of the action of enzyme catalyze found in raw chicken. In raw chicken livers, apparatus was set up as shown below. We have got these apparatus that are shown as that thermometer is shown there. We have got also our beaker, which has got the what? The chicken liver, and then obviously this is the we can see that this is the what? This is the the substance which is going to be put in, and then we are saying ice cold water bath 2 degrees Celsius, boiling water bath at 90 degrees Celsius, lukewater bath at 37 degrees Celsius, and then at each case we have got the same uh, substance being added there, as you can see. So formulate a hypothesis for this investigation. You know, hypothesis, you know what you need to understand is that a hypothesis it's a guess to an answer. I mean, it's a guess or an answer to an investigation. That when an investigation is to be done, we can preset the answer of that investigation. We can give the answer before the conclusion has been found. We can give an assumed answer to say the answer is what. So in this case, we can maybe give what you call a hypothesis to say that enzymes catalyze functions optimally at both. I mean, I can make to say that according to these various temperatures, I'm just going to say that enzyme. I have got various assumptions. I mean, hypothesis that I can give. One, I can say that the enzyme catalyze. I can say the enzyme catalyze the enzyme catalyst actually functions optimally at body temperature. It functions optimally at body temperature. I can say that of 37 degrees Celsius, which is maybe this one, which the mode. I'm saying enzyme catalase functions optimally at body temperature of 37 degrees Celsius. It functions optimally at body temperature of 37 degrees Celsius. I can also maybe say that the catalase becomes denatured by extreme high temperature. That is an assumption again. I can as well say that it becomes denatured or it is destroyed by extreme high temperatures, which is maybe that 190 degrees Celsius. I can also maybe say that I can as well say that the catalyst will be inactive in extremely low temperature, which is maybe 2 degrees Celsius. So the assumption, I mean the hypothesis is actually based on the various three settings that are there. It can work optimally at 37 degrees Celsius. It can be destroyed or denatured by extremely high temperatures, which will also which we can also say that its action will be negatively affected at 90 degrees Celsius. Or I can say that it becomes inactive at extremely low temperatures of 2 degrees Celsius. All those are different hypotheses that I can give on question one. And then the next question, in which test tube A, B, or C would the reaction take place? The fastest give a reason. Which one can we say the reaction will take place faster? The reaction will take place in which case either low temperature, boiling temperature, or lukewarm water. So in this case, we should understand that uh, the temperature, which is similar to human body temperature or more or less optimum to human body temperature, is 37 degrees Celsius. So I can say that in test tube C, which is the one which has got, I can say test tube C is actually the human body temperature where the enzyme can work best. Test tube C shows optimum human body temperature where the enzyme can work best. The enzyme can work the best. The enzyme can work the best. That is why I'm saying C, test tube C. Test tube C, which has got what? A more or less human body temperature. So that's what is the reason. That is the reason there. Question C. Which observation would indicate a positive reaction from the catalyst? Which observation can show which observation can show the normal reaction? So we can talk about in the sense where there is what? Where there is bubbles formed, where there is what? The formation of the bubbles. Observation would indicate a positive reaction from the catalyst in terms of, we are saying, the formation of the bubbles. Formation of what? Of bubbles is the one that can show what? that can indicate a positive reaction. And then D, what result would be observed after the experiment in test tubes A and B respectively. In test tube A and B, which you can see in test tube A and B, we are saying A is 2 degrees Celsius, A is what is 90 degrees Celsius. So what we are saying is what results will be observed in the two test tubes. So what you need to know is that that no bubbles would be formed in test tube A, no bubbles. No bubbles will be formed. No bubbles will be formed there. That is in A. Reason being what? Because was not broken down. Was not broken down. That is the observation that can be observed in A. And then, because obviously the temperature is too low, as you can see, 2 degrees Celsius. And then in terms of B, in terms of B also, we can also have no bubbles in B because of what? Because was not broken down because of high temperatures of 90 degrees Celsius. Extreme temperatures in B resulted in no bubbles also, no bubbles in B because the enzyme was denatured. Extreme temperatures destroyed the enzyme. I'm sure that we are talking about 90 degrees Celsius, that is shown there. The 90 degrees Celsius, that is, in this case, is too much. Therefore, 90 degrees Celsius, extreme temperature, 90 degrees Celsius, 90 degrees destroyed the enzyme, hence there is no bubbles again. And then E, name the property of the enzyme that is being investigated in this investigation. What is the name the property of the enzyme that is under investigation? As you can see, we are talking about temperatures and so forth there. So we are talking about enzymes, the sensitivity of enzymes to temperature. Sensitivity of enzymes to temperature. Sensitivity of enzymes to what? To temperature. Sensitivity of enzymes to temperature is what is being investigated in this case. And then name 2 factors that must be kept constant in this investigation. 2 factors that must be kept constant. We can talk about one, same amount of F1. We can talk about one, same amount of chicken livers. Same amount of chicken livers, because we need to see that they are not affected. I mean, the reactions, there is no variation to the investigation because the amount of liver was not the same amount of chicken livers. We can also talk about same concentration of enzyme used. same concentration of enzymes. Same concentration of the enzyme also used. Same concentration of the enzyme must be used. And also, number 3, same pH, same potential hydrogen or acidity or whatever potential hydrogen atoms in terms of the what? In terms of the chicken livers. The chicken livers must be exposed to the same pH. So give a reason why the investigation was repeated more than once. Repetition is always done to improve the reliability of the results. To improve reliability of results of investigation. Results of investigation. That is why we repeat the investigation. To improve reliability of results of investigation. That is what is supposed, that is exactly what happens there. The microscopes, the micrograms below shows 2 organelles that are shown there. Organelle 1 and 2. They are shown organelle 1 and 2. So we are given here organelle 1 and organelle 2. Organelle 1 and organelle 2. Identify them respectively. So you can see that 1 is what? 1 is mitochondria. One is mitochondrion. And then 2, obviously, you can see that this is a chloroplast. It's actually chloroplast 2. 2, you can see that is chloroplast. And then which one of the organelles shown above is found in a plant cell? I'm sure that you know that chloroplast is found in plant cell. Chloroplast is found in plant cell. Chloroplast is found in plant cell. Draw a labeled diagram of organelle 2. Draw a labeled diagram. I'm sure that this one, just for the interest of time, I can just tell you that you can just have to refer to your what? Please refer to notes this one. Refer to notes, please. You know, for me to start to draw that, you must just know that there is also the loop of the DNA. You should not talk about the lipid the lipid granule. You should also talk about all those functions that you know that they are found on the what? On the chloroplast structure. That is what is very important. But as long as I've shown you that organelle 2 is a chloroplast, just refer from your notes or refer to your notes that you can see the diagram that represents the chloroplast there. And then in which part of the organelle 2 is the pigment responsible for absorption of light? The absorption of light, it is actually done in the grana lamella. The grana lamella. I'm sure that we can agree together that that is the part of the chloroplast. I mean, is the pigment responsible for absorption of light. The grana lamella. And then 2.5 support the statement that says organelle 1 is called the powerhouse of the cell. Actually, because actually what is happening that is that the muscle cell carries out process of aerobic respiration, which is therefore breaking down the food breaking down food in the presence of oxygen, resulting in the cell storing what? Storing the energy, which we are saying the ATP. So the question is saying support the statement. So I can just say question 2.1.5, the muscle cell carries out a process of aerobic respiration. Muscle cell carries out, it carries out aerobic respiration, aerobic respiration. It carries out aerobic respiration, which by uh, breaking down food in the presence of oxygen, breaking down food in the presence of oxygen, in the presence of oxygen, which are resulting in cells what? Storing energy. Resulting in cells storing cells storing energy in the form of ATP. I'm sure that you know that energy is stored in the form of ATP. So that is the statement. Muscle cell carries out aerobic respiration by breaking down food in the presence of oxygen, resulting in the cells storing energy in the form of ATP. That is the thing that you need to understand. And then, which cell between muscle cell and the skin cell contains more of organelle 1? Explain your answer. It is the muscle cell. I'm sure that you remember that it is the muscle cell. It is the muscle cell. Reason because what is very active tissue is very active tissue. Because muscle cell, because it's very active tissue. That is the reason. Muscle cell, because it's a very active tissue class. Tabulate three difference between organelle 1 and 2. These guys, they just want you to draw a table. When they say tabulate, you must know that you should not do anything else. Just start by drawing a table. Just start by drawing a table, which we can say, I can say chloroplast, which is organelle 2, and then I can say also mitochondrion, which is organelle 1. So we are saying tabulate the differences. When we do this, we are actually tabulating. So one, the chloroplast is disc-shaped. It is disc-shaped. Also, this one is what is rod-shaped. It's rod-shaped too. We can talk about the site for photosynthesis chloroplast. The site for photosynthesis. It is the site for photosynthesis. Actually, it is site for what? Site for site for what? For cellular respiration. It is a site for cellular respiration. And then I can maybe add two other points. Fluid called the stroma. And then here it contains fluid called what? The matrix. I think last but not least, I think the last one that I can give is that it's long and this one is short. It's 4 to 10. And here it is actually about 1.5 is shorter, which is about 1.5. You understand? So these are the things that I can just give onto this one. And then last question is saying calculate the actual size. Calculate the actual size of the organelle tube in micrometers if the measured size of the image using a ruler is 86 millimeters and the electron microscope magnification is times 4,000. Calculate the what? It wants us to calculate. We must get it like that to say that when given this, we can say actual size is called. Does this question is question 2.2.8? 2.2.8. 2.2.8 is just actual size. Actual size, we say actual size is equals to measured size by ruler over the magnification. Actual size is equals to measured size by ruler over magnification, which is over magnification, and then which is given that we are given already that the measured size is what? It's 86 millimeters, which is given there. So we are saying 86 over what? Over our 4,000, which is equals to, we put it through a ruler, I mean our calculator, sorry, it goes to 0.0215 micrometers. So that is the actual size measured size by the ruler over the magnification, which is given there. So if you can do that, you can see that this is actually going to give you the what? The measurement, the actual measurement that we are talking about. Just quickly go to the next question. Supply the caption of this diagram. It is actually a sensory neuron that is shown there. This is a sensory neuron. Caption is just a title, sensory neuron. And then the question is saying, redraw a labeled diagram of the above structure and indicate with the arrows the direction of flow of an impulse. You need to know how does the impulse move, and you need to know that impulses moves from the dendrites to the synaptic nodes. So I can just quickly try to draw this diagram here. I can draw it here, just trying to what can I do? Let me just try to draw the diagram that I'm supposed to draw here. So we are saying sensory neuron is sensory neuron is our title there. And then the question wants us to draw the diagram, which is just simple. I'm just trying to draw the cell. I'm just trying to draw the diagram quickly. Obviously, you should know that this is the axon. We have got our cell body. You should know that at this side we have got we can say this side also is the dendrites. And then this side here is this, it's called the synaptic knob. And then obviously, which we can say that is the motor and plate side. Obviously, the direction of movement now should be showing that it's from the dendrites to the synaptic knob. So we have got the direction. We have the direction of movement, which is shown from the direction of movement. You can see that where the arrow is pointing, it's showing the direction of movement class. That is what you need to draw. You are actually just asked to draw the diagram of the sensory neuron, which is just like that, the axon which runs there, which carries the impulses, the nerve impulses, the cell body, which you can see there with the nucleus, and then the impulses have to transfer, move, and cross to to the synaptic knob, to the motor end plate. Let me do question next. I'm sure you can see that that's the central neuron and that's how it is like. Let me just go to the next question. Let us just go to the next question. Uh, from the diagram, identify a structure with the following provides nutrition and energy to the impulse. I'm sure that I have already shown you that it is the cell body. It is the cell body there, and then forms a synaptic cleft and transport impulses away from the neuron. You can see it's the motor end plate. I'm sure that we can agree and see that, as I've shown you here, the motor end plate is the end from where the impulses are ending. So it synapses with the what with the motor neuron. I mean with the yeah with it is the sensory neuron, and then it synapses with the interneuron. Sorry, the interneuron where it actually connects to the connector neuron there. So that is what is supposed to be understood, that the neurons, they lie like that adjacent to each other, but they cannot connect end to end. It's the histogram below, the composition of diets of two people, cereals that is unshaded, and then animal proteins, fruit or veggies, as well sugar and fats are shown there. And then from the histogram, from the histograms, name two types of food substances from diet B that represent a very similar percentage. In diet, we can say this is animal proteins and fats. Diet B, we are saying animal proteins, animal proteins and fats. Animal proteins and fats. And then give two functions of cereals in diet. They are actually a source of fiber. Cereals are a source of fiber, or we can also say that they are a source of energy. They are a source of fiber, they are a source of energy, and also source of roughage. Cereals, they provide all those. And then that is question B. And then calculate the diet difference between the percentages of fruits or vegetables in diet A and B. We have got between vegetables in diets A and B, vegetables which are there. So you can say that we are given that vegetables, we have got between A and B, it is 15, it is actually 18% here, 18%. And then here we can say that it is 15%. So that calculate the difference. Calculate the difference is what we can just say is 18% minus 15%, which is close to 3%. That is close to 3%. Which diet provides more energy? Give your reason. Which diet provides energy, more energy? I can say that from the diet that we are given there, we are given the diet A and diet B. So I'm saying which one provides more energy. I can say that It is diet A because it contains more sugar, fats, and proteins. Question D, A, because it contains more sugar, fats, or proteins. And then number E, diet B is eaten by a vegetarian. However, it is evident that this person also consumes dairy products, provide evidence from the graph. If diet B is eaten by vegetarian, however, it is evidence that this also person consumes dairy products, provide evidence from your answer, provide evidence there. Man, this is quite true. I mean, it's diet B, animal proteins, which is given here, animal proteins. So it is true that presence of animal protein in the diet, it is that which can show that it is because presence of animal proteins in the diet, because the diet contains, because the diet contains animal proteins. The diet contains animal proteins. That is the reason why we can say that it is also containing. That is the reason why that there is that reason there. And then define obesity. It's just excessive. It refers to excessive fatty deposits in the tissues and around the body organs. It's excessive fatty deposit. Excessive fatty deposits that is in the tissues and around the body organs. And around the body organs. Give one reason why obesity is regarded as a life-threatening condition. It leads to coronary heart diseases. That is the reason. It leads to coronary heart diseases. That is one reason. It leads to coronary heart diseases. And then it also causes high blood pressure. It causes high blood pressure. We can also talk about that. It is also, it causes depression. And also can lead to loss of life. It can also lead to loss of life. That is also quite obvious that it can also lead to loss of life. Diabetes is one of the sicknesses caused by obesity. Give the causes, ways of prevention and treatment of the disease. Question C, causes. Causes, we can talk about excessive consumption of carbohydrates. excessive consumption of carbohydrates, it can cause that, cause, we are saying excessive consumption of carbohydrates, carbohydrates rich foods, that is the thing. We can also talk about the it can also, because of the hormone insulin fails to convert excess glucose, fails to convert excess glucose. It can fail to convert excess glucose in the body. Excess glucose in the body. We can also talk about the treatment. One, it can be changing of change in diet. Change in diet. Two, it can also be insulin dose injection. Insulin dose injections. You can also have three. It can also eating food with less energy. With less kilojoules. That is four. We can also talk about regular exercising. All these are also very important in terms of uh, treatment of diabetes. I mean diabetes. And then we can go to the next question. The diagram below shows the dicotyledonous leaf. A dicotyledonous leaf that is shown uh, the give two functions of this plant organ. That's the first question. Give two functions. Give two functions of this plant. It's the leafing. One, it is important for gaseous exchange. Gaseous exchange is very important. We can talk about transpiration. We can talk about photosynthesis. Those are some of the processes that are very important: gaseous exchange, transpiration, and photosynthesis, which we can also give even also respiration. And then what is the collective name for parts labeled A and C? What is the collective name for parts labeled? What is the collective name for the parts labeled A and C? A and C, A and C, they are called what? Mesophyll tissues. mesophyll tissue. And then next question, name the metabolic process that takes place at B. The metabolic process that takes place at B. At B, we have got what? It is actually gaseous exchange. It's gaseous exchange, glass. That is 3.2.3. Is the metabolic process is gaseous exchange. And then identify the letter and name of the tissue with compact cells where photosynthesis takes place. Photosynthesis takes place in A. It takes place in A, which we are saying is the palisade mesophyll. Part A. The palisade mesophyll. That is the answer there where photosynthesis takes place. So I understand that it says the part and name. The part and name. So it is A. Photosynthesis takes place there. It takes place in A here class. The palisade mesophyll. That is A. The diagram below shows a part of a human skeleton. It shows part of the human skeleton, and then this is exactly part of a human skeleton, and then name the synovial joint that joins bone C to bone D and E 3.1 point, which joins C. It joins C to bone D and E. So it is joined by what? By hinge joint. It is the hinge joint there. And then give letters of the bones that make up the pectoral girdle. Give letters of the bones that make up the pectoral girdle. It is actually ABC. It is A, B, and C. Number question, the letters of the bones is A, B, and C. Provide labels for part numbered. Provide labels for parts numbered F and G. Labels for parts numbered F and G. F is actually here, as you can see, it's a metacarpal. It's the metacarpal here. And then G is the ligament. G is the ligament there. G is the ligament. F is the metacarpal. Write down the number of phalanges found in each hand. Number 3.34. It's actually the number of phalanges found in each hand. It is actually 14. Two sets of muscles are attached to the front and back of the humerus respectively. Name these two muscles. Here it is the biceps. The biceps. and the triceps. Name two substances that builds up and repair muscle tissues. It is what? Name the substance. It is proteins. I'm sure that you know that proteins is the building block of body. Describe how these muscles function to bring about movement. Question C. The muscles they work opposite to each other. They work opposite to each other in pairs, but with opposite effect to what? To each other. With opposite effect. And then what would be the consequences of if the muscle attached to the back of the humerus malfunctions? The person will not be able to lift the arm. Won't be able to lift the arm. Won't be able to lift the arm or carry any heavy load. That is what is happening. Lift the arm or carry any heavy load. I think the last question is the diagram undergoing phase in a cell division called mitosis. Identify the phase of mitosis represented above. As you can see, they are aligning at the equator, so hence we can see that this is metaphase. This is metaphase there. Give one reason for your answer. Chromosome line up on the equator, like I said. Line up on the equator. The chromosomes line up on the equator. Label parts A, B, and C. Label the parts A, B, and C. A, A is the spindle fiber. B is the chromosome. Actually, B is it's a chromatide. And then C is the central. You see? Spindle fiber, chromatide, central. Those are the letters. Name the phase that comes after one shown above. The phase that comes after this one, it's what is anaphase. And then how many chromosomes will be present in the cell are shown above at the end of mitosis. It's actually 2. It is actually showing 2 chromosomes. It's actually 2. As you can see there, how many chromosomes will be in the in the cell shown above at the end of mitosis is 2. And then what name is given to the abnormal and uncontrollable division of cells leading to formation of tumor? This is called cancer class. And then stage 3, biological importance of mitosis. It is actually mitosis, like I said before, it's important for growth. It is important for asexual reproduction, that is vegetative reproduction. It is important for replacement of worn out tissues. So these are the things there. Growth, asexual reproduction, replacement of worn out tissues are the two biological importance of what? Of mitosis. Thank you so much for following and tuning in. Don't forget to subscribe and share the content and like the work. And thank you so much for your time. Have a beautiful day. Look forward for the next presentation that is coming soon. Thank you.